Not all bacteria are alike. Some thrive on simple nutrients while others demand blood, chocolate, or bile salts just to grow. So how do microbiologists pick the right food for each pathogens? That's what this video is about, the 3 essential culture media that turn invisible bacteria into visible identifiable colonies. Culture media fall into 3 broad jobs. First, selective and differential media. They let one group of bacteria grow while flagging another. Second, enriched media. They add extra nutrients to support picky, fastidious organisms. And third, specialized media, designed for the most demanding bacteria that won't grow anywhere else. MacConkey Agar, blood agar, and chocolate agar, one for each job. Let's look at them one by one. MacConkey Agar is both selective and differential. Selective because it contains bile salts and crystal violet. These inhibit gram-positive bacteria, so only gram-negative rods grow. Differential because it contains lactose and a pH indicator called neutral red. Here's the part that matters. If a bacterium can ferment lactose, it produces acid, turning the colony bright pink to red. E. coli does that. If it can't, the colony stays colorless or transparent, like Salmonella or Xylella. In the lab, MacConkey Agar is the go-to for urine cultures. A pink colony on MacConkey almost certainly means E. coli, the most common cause of urinary tract infection, which is called UTI. Blood agar is an enriched medium, a nutrient base with 5% sterile C blood added. The blood provides extra growth factors so fastidious organisms like Streptococcus and Staphylococcus can thrive. But blood agar does more than feed bacteria. It also reveals how they destroy red blood cells. This is called hemolysis, and there are 3 patterns. Alpha hemolysis, a greenish discoloration around the colony, meaning partial lysis. Beta hemolysis, a clear transparent zone, meaning complete lysis. And gamma hemolysis, no change at all. In practice, a throat swab plated on blood agar showing beta hemolysis points straight to group A Streptococcus, the cause of strep throat. One plate, one clear answer. Chocolate agar looks like melted chocolate, but there is no chocolate in it. It's made by heating blood agar to about 80 degrees Celsius, which lyses the red blood cells and releases extracellular nutrients, especially Hemin called factor X and NAD called factor V. These 2 factors are essential for some of the most fastidious bacteria in medicine. Haemophilus influenza needs both factor X and V. Neisseria gonorrhea and Neisseria meningitis also grow here. So when a patient has suspected meningitis or epiglottitis, chocolate agar is the plate to use. It's the only routine medium that reliably grows Haemophilus, and that alone makes it indispensable. So here is how they stack up. MacConkey Agar, selective and differential, 4 gram-negative rods. Pink colonies means lactose fermenters like E. coli. Clear colonies means non-fermenters like Salmonella. Blood agar enriched for fastidious gram-positive organisms. The hemolysis patterns, alpha, beta, and gamma, tells you which Streptococcus you are dealing with. And chocolate agar specialized for the most demanding bacteria like Haemophilus and Neisseria. 3 plates, 3 jobs. Together, they cover the vast majority of clinical microbiology. The right medium turns a mystery into a diagnosis. MacConkey sorts by lactose fermentation. Blood agar reveals hemolysis. Chocolate agar fits the most fastidious. Next time you see a plate in the lab, you'll know exactly what it's telling you. Stay tuned.